Welcome to Genomics Gupshup, episode 53. Sorry about the long break that we took in between, but I wanted to welcome you back to Genomics Gupshup. This is an initiative by MapmyGenome to bring the community of genomics closer to you. And we do this by bringing together geneticists, doctors, nutritionists, fitness, and anybody else who has expertise in this area to you. This is not just available on YouTube, it is available on your favorite podcast, whether it is Spotify, Apple, Google, or anywhere else that you hear podcasts on. Today we have a very special guest. We have Dr. Ashwin Dalal. He's a pediatrician, but he's also a medical geneticist. He exemplifies excellence in medical geneticists, beginning with the prestigious accolade of a gold medal in his MBBS exams at Goa Medical College. He furthered his expertise by completing his MD in Pediatrics at Goa Medical College and then a DM in Medical Geneticists at the well-known SGPGI Lucknow. He is currently the staff scientist and the head of the diagnostic division at CDFD in Hyderabad, and he currently spearheads a state-of-the-art laboratory of rare diseases and is one of the most well-known figures in this space of genetics. I'm so glad and welcome Dr. Ashwin Dalal. So thank you so much, Dr. Ashwin. This is such a pleasure to have you. You are one of the most well-regarded and most respected geneticists in India. But you're not just a geneticist. I think you also have a degree in pediatrics.
Yes.
So can we first start off by saying where were you born and what inspired you to take this whole journey in becoming a doctor in the first place?
Yeah. So, I come from a small state of India, that's Goa. So I have been brought up in Goa, and so all my initial education was in Goa, and there was never a plan to be a geneticist. So
How could you study in Goa?
Yeah, Goa. Yeah, it's really nice place. I still long to go back someday. But yeah, so while studying, genetics was never on the plan. So my initial longing always was to become a computer engineer. So that was from beginning. Maybe I had my friends playing games, and I used to think how to build
Computer
games.
games.
Yeah, computer games. So this is talking of 1992, 91, those days. So yeah, but then at that time there were no entrance exams or something, so I had good marks in my 12th standard, and there was no exposure, I didn't know what to do, so there were only engineering and medical, so I didn't know what to do. My father's logic was engineering, you have to search for jobs, medical, you can give an injection and you'll get 10 rupees. So that was the logic. I went into MBBS. Yeah, but it was not like a great liking that time. But yeah, I completed MBBS, then got good marks. So that time it was all based on marks. There were no NEET exams or something. And at that time, people who got good marks were going to pediatrics. So I went into pediatrics. So it was not really kind of a choice or something planned or something. And yeah, so that way I did pediatrics. In 2002, I completed pediatrics. But tried doing private practice for some time, worked in a NICU, neonatal intensive care unit in Goa Medical College. So everything, all my studies are in Goa Medical College, and I was not really happy with what I was doing.
So how did this whole genetics thing come into?
Yeah, so at that time, that's what I was looking for. I didn't know, I didn't like. And at that time, actually, pediatricians further studies, the only option was neonatology. So I worked in neonatology for about a year, and at that time, we didn't have all these nice ventilators and all. And so seeing all those very small babies whom we really could not save, or they would go with a lot of complications. So I didn't like that. I tried private practice, I didn't like. So I didn't know really what to do, and at that time I had one of my friend, a classmate, who was doing endocrine surgery in this Sanjay Gandhi Postgraduate Institute in Lucknow. So one day I was talking to him, and he said, there are some pediatricians here who do something called genetics, and do you want to try? So I thought, okay, anyway, I was not very happy with pediatrics. I didn't have much options, so I said I'll go there. And so there was a call for interview for a non-DM senior resident. So it was not even a DM course, it was just for a job. I went there, and again, we had no knowledge much of genetics. We just have one chapter in our first year MBBS, and the pediatrics also, we would just staple those genetics pages, because even in our pediatrics training, we never had all those tests and all available that time. So we never used to read genetics. So I went there, and there also, now I didn't know, there was the first entrance exam that I gave in my life, and I didn't know what to read. And so my friend that time gave me a small book there from the library, and I started reading. And one day before I had gone for the exam, and whole night I was reading that book. So I don't know, somehow that night somehow I got hitched into genetics. I cleared the exam. I spent 6 months, and I knew I want to do genetics.
Which book was this?
This this is actually a small book which my guide Dr. Shubha Phadke. So they had just made a small book for they conduct a 15 days course in genetics every year. So that was just a small book made for that. But I don't know, somehow I liked it so much, this whole field, and then I spent 6 months there, and the whole institute and the environment, it was much different, much academic than what I had been in Goa, where you don't have that much of academics happening and all. So
that got you hooked.
That got me hooked like anything. I really found, this is something I want to do with my life, and never regretted it.
So SGPGI has, I think, probably the largest number of medical geneticists.
Yeah, at that time that was the only department where you could do a course, a formal course in medical genetics, a 3 years training course. So it has created almost so in the last 3-4 years now we have this same course in about 6-7 more centers, but that time it was the only center. So everyone used to come out of there, and then now we have people who have gone to different centers and created their own centers. So SGPGI was really a big influence on my life, and it really changed the approach towards maybe science or going into science. So even being a scientist also was not decided, because because when I went into genetics, that was the era of 2003 to 6, and although clinical genetics, a lot of that was there, and that was related to pediatrics, so it was much easier to relate to. But testing wise, we just had karyotyping, chromosomal analysis, Sanger sequencing. We had a single capillary Sanger sequencing that time in the department, and that too we were not allowed to touch. So we would just look at it from far away, think this is something big, a single capillary sequencer that time. So we had no idea. So that time genetics really did not seem to be very lucrative or something, but somehow I liked the whole concepts, and also that time genetics was just like academic specialty. It had not reached really the practice kind of thing. So
where did you all have like all the? I went to Lucknow and they said you must visit this Sharma ji ka Kichai ka whatever dukan, right? Was
yeah,
it
so
still
that,
there?
it's still there. So when I go back, we do make it a point to go there. It's nostalgic. It feels
So
nice.
where else do geneticists hang out? In Lucknow.
So SGPJ that time was almost 16 kilometers away from the main city. So yeah, we really didn't have much distractions that time, and no much mobiles and all. So we used to spend a lot of our time in library, especially. Now I don't find my students going to library at all. Everything is online available. But at that time, reading books was a really fun time. We enjoyed it a lot.
Yeah, I think that has changed completely.
Yeah, over
So,
the
but
years.
from Lucknow, then you came to
So-
Hyderabad, or how did you?
Yeah, so Hyderabad also was never, I just had heard the term Hyderabad, never thought that I would be coming. So actually after that, even when I joined, my guide there had told me, why do you want to join genetics? Like, it's not something lucrative, it's not as if you're going to go to a corporate hospital and start practice or something. But somehow I just liked it, and so after DM again there was a big question. So I went back to Goa and I didn't know what to do. Goa, though, there was nothing in genetics that I could do. Then, but I had given an interview in this CDFD. That too, one of my friends suggested that maybe just go there and give an interview. And till that time, like, never had been exposed to science or scientist or a scientific institution as such. I didn't even have an idea about. So in CDFD, it's not like the selection is more like you go there and give a talk. Now, I had nothing to give a talk on. Like, it's not like a PhD program where you work on one thing. We work on multiple different patients. So giving a talk there and all was really new experience. I just went there and first time I came to Hyderabad. That was a nice experience. I came 2006 or 2007 January actually. I came here, gave that talk, and I went back. I was not expecting anything to happen, so I completed my DM and then was going around medical colleges searching for jobs. I went almost 7 or 8 medical colleges in Mumbai and Pune, and they all said, "Yeah, it's very nice and all, but genetics we don't have any job for." So I had almost contemplated joining medical college in nearby, but so I just for the sake of it, I just gave a call to CDFD director that time, Dr. Gauri Shankar was there, and just I told him, is there any update, and I'm planning to join a medical college, and he said, no, wait, we are going to get back to you, and then they sent an offer letter as a scientist. So that was something again different because till then I had been trained as a clinician. So it was a big step, but then I thought anyway there was nothing great coming from the medical colleges also. So I joined CDFD, and again I don't regret it at all. It has been really good actually.
Today I think with the you've established a very significant center in CDFD, serving so many people who can't afford a regular test or any other. So I'm sure this whole journey of establishing that unit might have not been so easy.
Because as I said in that era, so when I joined CDFD also, it was really a good setup. It was much better setup than what I had seen in Lucknow, but it was not much of a clinical setup. So we had a research institute who was doing some amount of diagnostics. So there was Dr. Radha Ramadevi who was doing the OPD. There was Dr. Chandak had established the cytogenetics. But again, we were at very baseline level, like we were doing maybe karyotyping and a few PCR based tests. That was what was happening. But being in CDFT and with help from really, I am grateful for Dr. Gauri Shankar because he was also from a medical background. So maybe he understood that. So he never stopped me from experimenting or doing anything new. And so we slowly started building on these tests. So each time a new student will come, we would establish a test. So slowly we started doing MLPA, we started doing TP-PCR. Any new article on a diagnostics would come, we were able to establish it. But then again, there was a big problem about patients coming to a research institute. That was something which we all felt was not really good. And since I joined there, so DBT or Department of Biotechnology also helped us in doing a MOU with the Nizam's Institute of Medical Sciences. This is in 2008. We signed a MOU to establish the second department of the country of medical genetics. So the first was in Sanjay Gandhi, and the first in South India and second in country was at Nizam's Institute. So we signed a MOU. We established a department. Now it is almost 16 years since the department was established. We got faculty recruited there. Initial almost 10 years, the whole funding was done by CDFD to establish the department. Now it is a full-fledged, it is one of, between SGPGI and NIMS, they are almost equivalent departments. We have a DNB program, a 3-year program in medical genetics. We have MSc program in genetic counseling. We have a PhD program going on there. And last 16 years, it has been a great coordination where all the clinical work is done at the Nizam's Institute, all the laboratory tests are done at CDFD, and we jointly run this department. And so it has been a great experience and helped a lot of patients. Now, if you look at the number of patients coming there, it's almost 10,000 per year, which is a huge number for genetic specialty.
I agree. I mean, I think what you've been able to establish is a good way for many others to think about also in the future, right? Because there's going to be places where they are coming in, like government hospitals. But if we are able to get the benefit of the testing, I think it really helps them in taking it to the next level.
So we do a lot of training now. So we don't want it to be apex center based. That does not really help. So it has to be a lot of training at the grassroots level to enable them to do at least the basic tests, so that we have tiered kind of testing. We have the higher tests happening at bigger centers and the bare minimum tests at least happening. And there are a lot of government initiatives which are helping us to do that.
That's great. So, do you feel that there is, any change that has happened in the medical education level, like the curriculum, has it changed, or you think that we have a lot of work to do?
That still remains a problem. So, although there have been a lot of efforts, and we have been, as individuals as well as a society, that is Indian Academy of Medical Genetics, we have been writing to earlier Medical Council of India and now the National Medical Council to. But the problem is our undergraduate curriculum. There are so many things packed into that 4 years that it's difficult to add a lot of genetics there. But surely it is much better than what it was earlier. Now National Medical Council has made it a requirement that every medical college should have a medical genetics department. And the reason is very important, that medical genetics is not like an organ specific specialty. It applies to every specialty of medicine, whether it is cardiology, neurology, nephrology, everywhere there is a genetic component. And now slowly the textbooks also are adding a genetics part to every chapter. So that way slowly the medical teaching also is incorporating genetics, and we need more and more of this to happen, that it should not be that the medical geneticists do genetics. It has to be every clinician who knows a little bit of genetics and applies it to their specialty.
I think once they know what to do, then, or when to prescribe and what to do, I think it becomes easier for them to.
And it's not only the genetic diseases. Now we have a huge field of pharmacogenetics and all coming, which will apply to every specialty. So clinicians can no longer be away from genetics. They have to be aware of genetics.
I think even in my case, a lot of the medical societies are inviting me to talk about pharmacogenomics and other such areas, right? Even prevention. So I think it's slowly getting there, but I'm sure there is still a lot of work to be done to at least get that education to the, all the clinicians. But I'm glad that it is happening. I was watching one of your previous interviews, and you said almost 30% of pediatric cases will have some genetic component to it. Are 30% of the pediatricians becoming geneticists, or at least getting some knowledge in genetics, or how can we get, if that's 30% is a very significant number, how do we get more pediatricians, for instance, to be aware of these conditions?
If you look at all the specialties of medicine, basically the pediatricians and obstetricians are the most close to seeing genetic diseases, because many of these genetic diseases are seen in childhood and they have a prenatal onset. So it's the obstetricians who first notice this either as a fetus or they do prenatal diagnosis, and after the baby is born, it is usually the pediatricians who see it. So these are the two specialties which are very close, and a lot of efforts are being put to get these two specialties to do a little more of genetics in their practice. But we try to do it through different ways, like either through all these lectures. Now that's a good thing happening, that every conference does have a genetics lecture or something, which was not happening. Or earlier it would happen, but there would be very scant audience. It would only be the speakers and the would-be speakers who would be attending. But now we have seen a lot of conferences. The hall is packed for genetics, so that is really heartening. And I feel it's happening by need. I think everything happens by the need. Now they can no longer say that I don't need to know genetics. It is required that every pediatrician has to have some knowledge of genetics, because they are seeing these patients, and patients also are demanding that they want to do the test. Then if they do the test, the doctor has to know how to interpret the test. So slowly it's improving.
I mean, I don't know if this is published or not, but what percentage of people actually affected come and to somebody to see a genetic disorder? Do you think?
We won't have a number, but the problem is we always talk about that there are 350 million patients of rare diseases in the world, and of which by calculation 70 million or 7 crore are supposed to be in India, and of which 80% of the rare diseases are supposed to be genetic. So if you take 80%, by that way we have about 5.5 crore patients. But it's not that 5.5 crore are coming here. So even if the statistics are true, our reach has not yet reached the all the patients. We ourselves have not been able to reach. I feel, but that is a great amount has changed with the private sector coming into genetics. So till a lot of time, till 2000, maybe about 7, 8, or even after that, it was only the government centers who would be doing genetic testing, because that time genetic testing was limited to few tests and a very rare kind of specialty. However, slowly with the advent of newer technologies and the foray of private labs who have a much larger reach, so now even in small towns or villages, we have doctors ordering exome sequencing, which we won't have imagined if it was only limited to the government centers offering it. So I feel that has really changed the whole dimension of medical genetics in India, that now it has really reached nooks and corners. But the big problem now remains is the test is available, but the knowledge of interpretation is lacking, and that divide we need to somehow fill slowly by increasing more awareness.
I agree. I mean, most of the time, when we see it, we see mostly either it's the geneticists or the genetic counselors or others who are able to then understand, interpret, and then help the patients as well. The challenge will be that, there is a need for better quality of family history or understanding, which sometimes, at least in our case, we don't get a lot, and we have to again constantly keep asking for that. Can you maybe tell a little bit on the importance of the quality of the understanding of the history, but also the quality of the data that you get in some of these bigger tests, right? Like there is obviously you do a lot of work on tests where are very specifically things like thalassemia and others.
Yeah.
But on the exome sequencing, for instance, now you did mention that private industry has done a good job of spreading the awareness. But I think we are also getting to a point where we are maybe spreading too fast without
that.
And do you think that there is importance in understanding that what are we really looking for in these tests for the doctors, right? Like when they are finding, one could be that they look at the patient, they know it is a problem, or it could be that the patient says, I want a diagnosis. Where do you see that the value of high quality data and the value of interpretation comes for the progress of this field?
Yeah, so it's really from both sides. I think it's from the referring clinician as well as the laboratory. So it's very important that the referring clinician provides the full phenotypic information to the lab, because the doctor is seeing the patient. The lab has no idea about what that patient is having. So the lab is totally relying on what information has been given by the doctor. And so if you don't provide enough information, when you do such tests like exome sequencing or genome sequencing, you have thousands of lakhs of variants which are present. Now to narrow down to that one disease causing variant without a proper phenotype, it becomes very difficult for the lab. So it's very important, I feel, that the clinicians provide a good clinical details to the laboratory. We have had so many examples of this happening that we had one patient who was sent by a doctor to the laboratory saying he has difficulty in getting up from sitting position. Now, difficulty in getting up from sitting position is typically a muscular disorder. So the lab has just looked for the muscular disorders and they have reported a variant of uncertain significance in one muscular disorder. Now this patient then came to one of my colleague and so he is a clinical geneticist. So on seeing the patient, he knew that this patient has another disease phenotype like Morquio syndrome. Morquio syndrome is a mucopolysaccharidosis disease where the patient will have difficulty in getting up because it affects their hip joints. So then he told the laboratory back again to look at variants in the Morquio syndrome gene, and there was a pathogenic variant homozygous there. So if the information is not given to the lab, then the lab doesn't know where to look for. And they would, even if something is there, they would not report because it is not matching the phenotype. The genotype phenotype match is the most important in any genetic diagnosis. So here the lack of knowledge between the laboratory and the clinician creates this problem where then the clinician is going to blame the lab saying that you did not report, and the lab will say that you didn't give enough information. So I think that is what the role of clinician is always to provide. And this is not only for genetic tests. I feel this is true even for a pathology test, a microbiology or maybe a radiology test also. The more clinical information you provide, the better yield you will get for the test. Now it is also a problem, as you said, from the laboratory point of view, because in India we don't have such kind of a regulation of who can do a particular test. And so like any other new technology, genetics also is a new technology, so there is sudden explosion. So we have so many labs coming up, and the quality of data is a issue when you see across different labs. So if you do the similar test from different labs and you look at the raw data, you would have different kind of quality of data. Even if the data is same, the interpretation also becomes important, because this genetic test is not something like a blood glucose where you put the blood and you get a
The machine
value.
is not giving.
Yeah, the machine just gives you the data, but the interpretation is always by the expertise of uh, the clinical geneticist or the counselor who sees the reports and all. So I think, but that is like a bubble which happens. See, when even a MRI came, it was suddenly like every headache people would want to do a MRI. Similarly, now every patient just goes for exome sequencing, but it's not really needed. If you have a small gene or a one mutation causing disease in all patients, like sickle cell anemia or achondroplasia or Apert syndrome, these are diseases where one mutation is known to cause disease in all patients of the world. A simple Sanger sequencing of maybe 1000-2000 rupees will give you the diagnosis. You don't need to do an exome. So this awareness slowly needs to increase about. Again, here the demand has to come from the clinician for asking for a good quality report. So I think it averages out over time. Over time, the good players will remain, the low quality will get out. But we are now in a fluid state where so many things are available. So you have exome, you have genome, and now you have transcriptome coming, you have methylome coming. The doctors are educated more by the medical representatives going to them rather than maybe a academic thing. So I think this always happens with any new technology, but then slowly it levels out, and then you have.
It will eventually will get there. But one of my worries is that sometimes we race to the bottom, and as an industry we need to be aware that there is quality that makes diagnosis, and when you get the right diagnosis for the patient, then you are getting the best. So I think ultimately we all have to self-regulate also if we can.
Yeah.
And of course there's NABL, there is CAP and others that are there, but I think there is work to be done, I guess, from all sides.
That's right. Yeah.
So you spoke a little bit about pharmacogenomics, and I remember you gave a very nice example in one of your videos. Can you maybe explain what pharmacogenomics and the carbon design? You said there are some cases of.
So Pharmacogenetics actually, although we have coined this term now, it's really not a new concept. We have been doing this in medical practice earlier, that we have a lot of cases of malaria. So malaria, the Plasmodium vivax, which is the most common type of malaria. This here we used to give a drug called as primaquine. Now, primaquine is a drug which, if the patient is a G6PD deficient patient, a glucose 6 phosphate dehydrogenase enzyme deficient patient, then he gets severe hemolysis, his blood cells break down, he will get renal failure. So, we always used to do the G6PD test before giving primaquine. This is an age old concept, but that time we didn't have this word pharmacogenetic, but that is what we were doing. We were measuring an enzyme and then giving the drug depending on that. That slowly came to more scientific way, and a very good example that I like to give whenever we talk is about 6-mercaptopurine. So 6-mercaptopurine is a medicine that is used to treat childhood onset leukemia or blood cancer. So when this medicine is given in our body, we have an enzyme called as TPMT, that is thiopurine methyl transferase. This is the enzyme which metabolizes this drug, and then this drug is excreted in urine. Now, what is seen in population is that about 88% of the people have a good level of this enzyme. In normal people, 11% of the people have lower levels of this enzyme, and 1% of people don't have any activity of this enzyme. Now, this enzyme is very important for the drug metabolism, but it does not cause any problem if you don't have that enzyme. So now when you give this drug 6-mercaptopurine, those who have good level like the 88% people, they will respond to the drug, their leukemia will get controlled and everybody is happy. The 11% of people who have a lower activity of this enzyme, because this drug now remains in their body for a long time, they will get a lot of side effects. And this 1% of people, they will die because of the drug rather than because of the disease. So now if we can test these enzyme levels and we give normal dose of drug to the 88%, we give lower dose of drug to the 11%, and we don't give the drug to that 1%, then all 100 can be happy. And that is what is the field of pharmacogenetics, where you do a genetic test and find out what is the susceptibility of this patient with this disease for this drug. And this is seen across all the drugs that we prescribe in medicine, whether it is antihypertensives, coronary artery disease, any drug, we see that all patients don't respond in the same way. Some respond in a very good way, but in routine medicine what we do is that we just change the drug. But here, if we have a logical way of doing a test and knowing who will respond better, who is not likely to respond, so give him another drug. That is what is the field of pharmacogenetics, and I feel the future of medicine is going to be pharmacogenetics. It's not yet legal in India, but legally in other countries like USA,
It is legal, it is not mandatory.
Yeah, it's not mandatory, it's not regulated. But in US now you can't prescribe certain drugs, like there's a drug called carbamazepine. It is used to treat seizures. Now, the carbamazepine in few people can result in a very severe condition called as Stevens-Johnson syndrome. It's a very severe condition. If you have seen those people, whole skin, big boils come up. And just because that drug was given. So in US it is mandatory to do that test and find those people who are likely to develop Stevens-Johnson syndrome, and then only you can give that drug. And this is going to become true for almost all the drugs that are prescribed. And so in future every clinician will have to do that particular test. So what we say now is that basically you sequence the whole genome of an individual at one time, but read it again and again. So every time you go to a doctor, they would put that maybe like a credit card or something, you have like a chip, you put in the drug and you will get the information what is the susceptibility, because the genome is not going to change from birth to death, it remains same. So you sequence once, but you read it again and again for different aspects that you want to see. So whether it is pharmacogenetics, whether it is susceptibility to certain diseases, so you can read it again and again and get the same information.
I absolutely agree. I think we are seeing a big uptake on the pharmacogenomics side. But I often tell people that don't prescribe anything yourself, go to your doctor, because it's not just the pharmacogenomics that you'll get, there are other elements that only your doctor can understand. So let's move on. You've done a lot of research work, right? Which is the one you're most proud of? I know that you have recently won on Serpin, and then you have on other things as well.
So research also, as I said, was not a planned thing. I have always been trained like a doctor, but then being in CDFD really gave me that opportunity to go into research. And like I didn't really plan onto research, but some of the initial patients actually got me to get into research. And a very nice example is of a condition called as pseudorheumatoid arthropathy of childhood. That was a very nice experience in my early days when I had just joined.
You were the first to come up with it.
So basically this all began with one patient who came to us. So he had recently joined a government job, and so this patient came to us saying that he wants to know whether his disease will go into his child. So that was his question, because he had got a government job and he had recently immediately got married, and now he was suffering. So what he had a problem was something where the joints were swollen right from childhood. So it was swelling of all the multiple joints of the fingers, the knees, and his mother was similarly affected. And so so both mother and child affected, it would look like a autosomal dominant disorder. And in which case you would say, oh my god, then his children have a 50% chance of having this disease. So when he came to us, again the first thing always in genetics begins with diagnosis. Without a diagnosis, we can't do treatment, we cannot do prenatal diagnosis, we can't help the patient in any way. So reaching the correct diagnosis is very important. So that time we saw his phenotype. The phenotype looked very much like rheumatoid arthritis. We all know about rheumatoid arthritis, very common, and then his mother was being treated for rheumatoid arthritis with steroids, and she had got all side effects of long-term steroids like cataract, like fat deposition. So she was all suffering more from the side effects of the steroid now. She had a totally bent spine at that time. So we checked this patient, and it's not something how a clinical feature of rheumatoid arthritis would look. So we did a lot of reading, and then we found just few months before a gene had been described to cause this condition called as pseudorheumatoid arthropathy of childhood. So it's pseudorheumatoid. It looks like rheumatoid arthritis, but it is not rheumatoid. It's not autoimmune. So then we thought, let us check in this patient, and that's a recessive disorder. That's not autosomal dominant condition. So we found a homozygous mutation in this patient, which was surprising because we were thinking of a dominant mode of inheritance, but this was a homozygous mutation, which is a recessive. So then we went and checked the mother. She was also homozygous, and this happened because the mother's parents were consanguineously married, and she was also consanguineously married, and so her husband was a carrier, and that's how this son also got the disease. So we now know that this is an autosomal recessive disorder. So both the copies of the gene are affected, then only the disease will happen. So we checked his wife, who was now not consanguineously married, and wife was not having any mutation in that gene, which meant that his all children will get just one copy and not the other copy from his wife. So he had no risk of getting any child with as many children as he can have. He did not need prenatal diagnosis, he had no risk of having a child, although his mother and he was suffering. So that was something really interesting which came out of testing. Otherwise, we would have told there is a 50% chance for you to have a child. Now we could tell him, none of your children will be affected, just go ahead and have children. And so then we were happy, okay, we did this diagnosis. But I don't know, somehow it happens that when you see one patient, then you publish it, and then people start sending you cases. And so we got a few more cases, about 5 cases, and again we did this mutation. And so at that time in India, this was the first time we had described this disease. We got more patients, we started working on it, and slowly it went on to so much that we had 100 patients of this disease. It was a huge number. And then we wrote gene reviews on this particular disease. So gene reviews is maintained as a review for each disease by NCBI from the National Institute of Health in USA. And so that was a very nice experience to go from one patient to finding 100 patients of this condition, and that really hooked me on to going into more research, going into finding all the mutation spectrum of diseases. And then we had the advent of these new technologies like exome sequencing, and that really opened our gates to identifying novel genes. So earlier we had recently reviewed and found that more than 50 new genes have been identified by scientists in USA using Indian patients. Because we did not have all the technology or the wherewithal to do these new gene identification kind of tests, we would always be sending samples abroad and they would do the checking and they would describe a new gene. But now with availability of these technologies, now we had that capability of identifying new genes. So we entered into this field in about 2011 or 12. We started doing exome sequencing, and it was really a learning experience because a lot of computational thing was involved, because it was not like Sanger where you could visually see with your naked eyes. You needed a lot of computation, and maybe somehow my wish of going into computer sciences helped me. I got hitched on to it into data analysis. I liked it. I started doing it, and we identified a first novel gene in 2010 using a patient which my guide Dr. Shubha had seen in early 90s, and we had just kept it as a novel syndrome. Now we called that patient back. Now the patient was an adult, and we identified a novel gene called as BHLHA9, which causes a finger abnormality. And that was really heartening to see that we can also do what is being done abroad. And then we ended up getting almost 7 to 8 new genes till now. And recently we had the new gene, that is SERPINA11, which was identified by our group in 2 fetuses who had this mutation in this gene. So everything starts with identification of a new gene, because we have about 19,500 genes in our each cell, but we know diseases only for about 7,000 or 7,500. 13,000 genes, we still don't know what disease they will cause if there is a mutation. And these are waiting to be described by maybe our future generation of students and young scientists who will be coming into this field. So I'm really optimistic that now we really can't complain that something is not available. Everything is available. Every test is available in our country. We don't have to send a single sample abroad to do any diagnostic test. All facilities for doing novel gene identification or their functional characterization, everything is available. We even could go up to mouse model development for the recent gene that we did. So I think it's really a great time to be in this field at present.
I agree. I think, and it's great to see how it, from a patient, you've been able to create, a community rather, right?
Yeah.
Because from one patient to many patients, I
Yeah.
think other patients who are looking for that also can find.
That's right.
So if today there's some patient in some village of India, how do they finally come to CDFD? Is there some process, or it is coming through their doctors, or?
That's something where we are lacking, that reaching that patient. So because medical genetics is such a specialty, it's a referral specialty. Usually nobody walks into a medical geneticist. Similar to what would happen earlier, nobody would walk into a cardiologist. Maybe a medicine specialist would refer to a cardiologist, but now people would just walk into a cardiologist. So maybe time will come in future where people may come into a medical genetics clinic, but as of now we depend a lot on the primary care physician who has to pick up these patients and then refer to a specialty. So there it becomes very important, as you said earlier, about spreading awareness among the clinicians, so that at least they pick up these cases and refer. That is very important. Otherwise, a lot of these patients will remain in the community without a diagnosis. Nobody knows why they got the disease, why they died, and it just the disease perpetuates in the families. So I think that last mile is something what we are lacking. As I said, everything is available, but that everything is available in the higher centers, in the cities, and slowly, but it is going across, and that's what I said. I am really happy and thankful to the private laboratories who have helped to take it to the masses. So now these tests are becoming available, awareness is there, and so I think slowly we will be able to reach that last mile in near future.
I hope so too. I think there is that need that is there, but at least we have come to a point where from the times when, for instance, when you started or when I
Right.
started, I think things have changed quite drastically. So
Sure.
I think in the next few years we'll get
Yeah.
there. The other thing I found really fascinating in your research that you have done is that you also created a consortium and published a lot of those exomes that was done
Yeah.
a few years ago. So why did you feel the need for doing
Yeah.
that? I know it's important and we are very
Yeah.
thankful that you're doing that. But maybe it's important for others to also know why it's important for us to publish or tell people what.
Yeah, so that initiative also is out of need, so we didn't really plan for it. So as we know, when we do exome sequencing, we sequence all the coding region of the genome, which is only 1% of the genome. Our genome is 3.2 billion bases of ATGC, of which only 30 million are the coding region, and that is what we sequence in the exome. And when we sequence 30 million base pairs, we get about 30,000 variants or what we say variations among individuals. Now most of them are the polymorphisms. So these are non-disease causing variants which make us all different from each other. If you compare our genome with a chimpanzee genome, we are 99% same. So we have a lot of monkey inside us. If you compare two individuals, we are 99.9% same. Just that 0.1% difference makes us all different from each other, but that 0.1% is also millions of bases, and if you look at the exome, it is thousands of bases. So now we have to remove all these bases which are different from each other. They don't cause any disease, they just make us all different from each other. These we call as polymorphisms. Now these polymorphisms are different in different populations. So when we do a exome sequencing, we come across about 30,000 variants. We do filtering of all those polymorphisms, and then we reach about 1000 or 2000 variants. For doing this filtering, we need normal population data. We need to know what is normal. So we consider anything that is more than 1% in the community as non-disease causing and likely to be normal. This is especially for the rare diseases. Now, problem is that we have a lot of databases which were made in the Caucasian population. So we have the 1000 genome database, the NOMAD, which has now more than 8 lakh individuals, but they are mostly of Caucasian origin, and we have very less contribution from India. And so the problem happens that when we remove these variants, if there is a Caucasian patient, from 30,000 variants, you will come down to maybe 300 or 400 variants. In Indian patient, if you do the same filtering, you will end up with about 1000 to 2000 variants, because those Indian polymorphisms are not yet in that database. So that was causing a lot of problem for us to do this kind of analysis. So that time, because we didn't have any Indian database, we thought let us put together all our exomes. So from our center, from SGPGI Lucknow, from AIMS New Delhi, and from KMC Manipal, we put together a total of about 1500 exomes and created, that was one of the first Indian databases that we created in 2021, and we published it and we put it online on our website for everyone to make use of. And we ourselves found that a lot of variants now would get removed because they were Indian polymorphisms. So it was really helpful. And after that, there was a big initiative from Government of India, what we call as the Genome India, wherein 10,000 normal Indians have been sequenced. And recently, in last January itself, 2025, the Prime Minister has released this data, and hopefully slowly it will get available to all the laboratories to make use of. That will reduce a lot of effort that has to be put to identify that single variant in the patient. And For India, we need a much larger, so the 10,000 is again a very small number for such a big country like India where we have 150 billion people. But slowly, probably more and more of these databases will come, and then it will make life easy for identifying these variants. But these databases are very important, and this data needs to be made available to all the community, just like we are using the 1000 genome or the nomad, and that will make things much easier in the long run.
No, I can't agree more. I think part of why even I started was that there was less representation. Can we get started? So I think, when we were talking and we realized that for certain types of genes, you manage to attract more patients of that kind. I think it would also be helpful for some of the private industry, at least for the rare disease cases, because it's so hard to find that community. That if we can build a more stronger, cases, I think it will be beneficial for the rest of the country as well. Right? Okay. So I know we've actually gone. I don't know whether it is almost an hour or more, but we have the interesting part, many interesting parts of the section still there. So if you want to take a small break, it's fine, or we can continue. So. What are your thoughts? I mean, you've taken us through your career. You were a doctor who then pediatrician who became a geneticist. What would you tell the younger generation today in terms of advice? What should they be doing? I know you mentioned that genetics is a great field, and it will be there in every hospital. So any advice that you would want to give them?
Yeah, so we do have a lot more people now getting interested into genetics, as is seen from the number of people who are trying to get into this field for the trainings that are being conducted. So that awareness has surely increased, and to the new students who are trying to get into this field, I would say medical genetics is going to be the future of medicine, so there is no doubt about it. And it is going to affect our medical practice in a lot of different ways. So, one of which is the most low hanging fruit is the rare diseases, where a medical geneticist or a clinical geneticist is important to identify them. Now, newer therapies are coming, so it is becoming more and more important that we identify these patients because therapies are becoming available. So that is one part, but a lot of major part is going to come with the advent of the pharmacogenetics, and we are going to need an army of people to interpret these results, because this pharmacogenetic testing is going to bombard the clinicians with so many variants, and how to interpret those variants and how to inform the patient about this variant. So we are going to need people trained at all different levels, right from the clinical geneticists to the genetic counselors to the laboratory technicians. At all different levels, there is going to be a large need for people trained in this field. So I feel there is a bright future in this particular field, and we do encourage anyone who is interested. A lot of students come during their MSc or during their PhD to us to get trained in this field. So I feel things are going to be much better in future, and there is going to be a large need for people trained in this specialty.
That's great. So you mentioned, SGPGI is where it started, most of the medical geneticists. But I know we got someone who graduated from AIMS. Where are the other places that they can do?
So the DM in So DM in genetics, DM is a 3-year medical course after MD in any of the medical specialties. So after MD, DM can be done. The first was Sanjay Gandhi Postgraduate Institute in Lucknow. After that, the same course is available now in AIMS New Delhi, in Postgraduate Institute in Chandigarh, in AIMS Jodhpur, then in Kasturba Medical College Manipal, and then we have a parallel DNB program which is equivalent to the DM, that is available in Nizam's Institute of Medical Sciences in Hyderabad and at Sir Gangaram Hospital in New Delhi. So across the country we have about 7 centers now where DM can be done. So that is from the DM point of view. Then we also need the genetic counselors. So we have a MSc program in genetic counseling, which again has been started in NIMS and in SGPGI Lucknow and in AIMS New Delhi. And I hope in KMC Manipal also they have. So I hope more such programs will start, because as I said, we are going to need people trained at all different levels. So genetic counselors especially are very much needed. Almost all corporate hospitals now want to have a genetic counselor. All laboratories need to have a genetic counselor, because to talk to the patient, the pre-test counseling and the post-test counseling is very important in a genetic test. And doctors don't have enough time to do that. So genetic counselors become an integral part of this specialty, and they are really going to play a big role in the future.
Okay. And do you think that there's a message that you have for families who might be affected by genetics? I mean, everybody is, but
Yeah.
at least for people where it can result in a real serious disorder. You mentioned about consanguinity
Yeah.
in the first case that you found. So what would you advise people who are planning a baby or planning to get married?
Yeah, so we call it something called as a preconception counseling. So earlier we started by doing diagnosis of patient after the patient is already having a disease, and that time what we could offer them was only a prenatal diagnosis to prevent birth of another child with the same disease in the family. Now that is something called as secondary prevention, but the first child is already affected. What we would like now to do is to prevent the birth of that first child also who is affected. That is what we call primary prevention. And so preventing that first child being affected, we can do it only if the couple comes to us before conception. In some cases or in some countries, people do premarital counseling. We really don't encourage it so much, because we don't want people to decide whom to marry based on a medical thing. That has to be more of a personal decision.
They can decide to get married, but they should
Yeah.
know what the consequences are.
Yeah, but then the problem is of privacy, the problem is of stigma,
Okay.
that if you know you have a particular disease, it's becoming, geneticists will make someone like a manglik or something, where you have something which prevents you from getting married. So that's why we prefer to do a preconception, that means post marriage but before planning for a child. That is really the ideal time where a couple should consult a genetic counselor or a clinical geneticist. And what we would do in a preconception counseling is really to help the couple to understand that we don't think about it, but every couple who gets pregnant, they take a risk of having a child with abnormality, because we call it as the background risk or the population risk. And this risk is about 2 to 3%. That means every 100 couples who marry and have a child, 2 to 3 of them will have a child with some or the other problem. And this 2 to 3% is through USA, Africa, wherever you take. It's a problem of human reproduction. But we don't think about this particular risk when we plan for a pregnancy. We always plan that we are going to get a normal child. Now, the primary prevention is going to help us to take care of at least some of these 2 to 3%. If we can do certain tests and find out if the couple is at risk of having a particular child with a particular disease, then we can prevent this from happening. And so this primary prevention, basically during the preconception counseling, we explain the parents that we can do carrier screening for some of the common genetic disorders. I said there are 7,000 genetic diseases. Very difficult to screen everybody for all diseases. We can do it, but it's going to be expensive with a lot of problems associated with that. So as a society, the Indian Academy of Medical Genetics, along with the Society for Fetal Medicine and the Federation of Obstetrics and Gynecology, has given out guidelines that all couples who are planning for a pregnancy should do carrier screening for beta thalassemia and spinal muscular atrophy. These are two important diseases which can be easily prevented if we do carrier screening and then do a prenatal diagnosis in those where both the parents are carriers. So by doing this, we can do a primary prevention of these two conditions. The second thing that we do for primary prevention is to do prenatal diagnosis of Down syndrome and neural tube defects. So if we take care of beta thalassemia, spinal muscular atrophy, Down syndrome and neural tube defects, a vast majority of genetic diseases would get tackled.
70-80%?
Almost, because the others are all ultra rare disorders. But then again, these disorders change according to community, because in Caucasian population, if you say, they have cystic fibrosis more common, they don't have thalassemia more so common. In India, that is why we need local guidelines for all these conditions. So that is why we came out with these guidelines. So we try and see that every couple who comes to us who is planning for a pregnancy, we do the carrier screening before planning pregnancy for these two conditions, and during pregnancy we screen them for neural tube defects or any other malformations by ultrasonography and a Down syndrome screening test. So this is the basic thing. If that is we are able to provide it to all the couples who are undergoing who are planning for a pregnancy, then we could make a difference to a large extent to prevent these conditions.
I think that's also, it makes the cost for the parents also much lower if you're
Correct.
just looking at these specific cases. And maybe if government centers are doing it, then people who
Yes.
can't afford can also do it. And I
Yeah.
think that will help in at least reducing the burden that we have today of rare
Correct.
diseases. So 70-80% is majority of the,
Yeah.
a lot that
Yeah.
we can solve. And then the rest, I think we can.
Yeah,
That
so
will
the
be-
rest will need a more focused approach, because we can't look at all the conditions in all patients, because it becomes difficult to interpret. We don't still understand all the variants. A lot of variants of uncertain significance will come up. Interpreting them becomes difficult. So we don't advise everyone to do a exome sequencing in their preconception counseling. But if there is a family history or if there is consanguinity, then we advise them to undergo these carrier screening for all other conditions also, because consanguinity can make a person more susceptible to develop these rare diseases. But again, consanguinity per se, we don't advise that you ban consanguinity, or because it's a social custom and it has survived over generations. And that also has a reason, because as I said, there is a 2 to 3% probability that a couple who is non-consanguineous would get a child. A consanguineous couple, that probability goes to about 5 to 6%. So the risk doubles. But even in consanguineous parents, 94% of them have a normal child. So that is why this custom has survived, because it's a social custom. So as a community, we don't really advise to ban consanguinity or that consanguinity is bad or something, because that is what is being followed in the community. What we advise is that if there is consanguinity, they approach a clinical geneticist and do a test, so that they know if both are carrier for a particular disease, we can do prenatal diagnosis for them.
I think that's great. I think it's important for us to tackle the problems first, the bigger problems,
Correct.
and then go
Yeah.
into that. So now we'll come to the part which is the fun, the rapid fire part.
Okay.
Alright. So, so if you would not be a geneticist, I mean you're a pediatrician,
Yeah.
so then what would you be?
Yeah, as I said, I would have been a computer engineer sitting somewhere.
You're already doing that.
I wanted to do that, and I somehow get to do that a lot with newer technologies. So that's why maybe I'm happy doing genetics and
And
some of what I wanted to do. Yeah.
So tea or coffee, what is fueling your research?
I like tea, but I have a very nice anecdote for coffee actually. So we had one patient who was referred to us from Bangalore. My colleague referred to him. So he had a problem that he had a lot of abnormal movements of his body. Right from childhood, he had these involuntary movements. We call them as paroxysmal movement disorder. So we did an exome sequencing in this patient, and we found one mutation in a gene called as ADCY5. This is the adenyl cyclase enzyme. And so at that time we just gave the report. But when we were reading about this condition, we found a very nice case in Madagascar. There was a family with this same problem. So that patient, what he went and told his doctors is that he has this abnormal movements, but when he goes and drinks coffee, these movements go off. So that was quite interesting. And then when he went to the doctors were also surprised. They felt he's just talking as a coincidence. So then they had another patient. So they did what they call
Coffee cream.
yeah. So they took this patient and they admitted him and gave him coffee, and then the movement stopped. And then they said, okay, what is stopping this movement? So they gave him decaf coffee, where the caffeine is removed, and it did not act. So caffeine was somehow stopping these movements, and when they looked up, they saw that so there is the adenosine which binds to this receptor, and caffeine has a very similar structure to adenosine. So what was happening was adenosine was binding and making this enzyme more active, and then causing all these movements. When you give caffeine, it was dislodging the adenosine, and caffeine was going and sitting there, and the movements were stopping. So we gave our patient also coffee, so we did not know how to give him caffeine. So we found those sunrise sachets, and the dose that you have to give is much larger. And all of our coffee has this chicory in it, which is very bitter. So and we had to tell him every time one dose is that full packet of. It's not like you drink milk and coffee. It was that whole packet of coffee that he had to take. But his movements totally disappeared, and that got then published in the papers and all. But it was really nice experience for us. But then we had a problem of what to do for him. He every day taking so much of coffee was difficult. Then we tried getting caffeine tablets. That is not available in India at that time. So
Now you do.
we imported caffeine for him. This is, I think, I'm talking of 2013-14.
Wow.
And so we imported, we got some of our friends who were coming from US to bring caffeine tablets, and that made his life little easier. He is now taking coffee, and his movements are not there. He's so happy, like he's doing cycling, he's doing swimming,
Oh,
which he
that's
could
great.
not do earlier. So it was a nice anecdote with coffee. When you ask
can
coffee,
also realize-
coffee could
Yeah.
treat patients. So that was nice experience.
That's a fantastic, I think, a great way of like connecting the genetics and coffee.
Yeah.
also. So if you could have dinner with any scientist, past, present, anybody, who would that be?
I feel Gregor Mendel. My whole life revolves
So you
around
have
single gene diseases.
You
So
have pea soup?
pea soup with him, yeah, maybe. But yeah, but that's another nice experience that we had. So 2-3 years back, I had gone for this European Society of Human Genetics, and that was the 200th year of Gregor Mendel's birthday. And so these people, they have this lot of ancestry information. They had information of where Mendel was buried. So they dug up his grave and found the bones probably of Mendel. And now they had one book which was written by Mendel, and in that they found a strand of hair which they believed may be of Mendel. And the power of genetic testing, you can see they got the DNA from that hair and they matched it with the DNA from those bones, which
They matched?
and they matched, and it was it was shown by them in that this, because I had gone to Austria and Mendel was from Austria, so that lab there had done it. So it was really surprising. After hundreds of years, we could get DNA out of that and match it. But yeah, Mendel is something who opened
So
up
what
the
did they
field.
find anything in his ancestry also?
Those were small strands of DNA. But maybe they might work more on it. But yeah, Mendel has, we call all our diseases Mendelian disorders.
That's good. So what is this one, if there is, a myth on genetics that you wish people would stop believing in?
Yeah, that really is a problem. What clinicians or even bureaucrats would tell us that even if you diagnose a disease, what can you do about it? So I feel there is a lot of hope for even patients with genetic diseases. If not something, getting the diagnosis also is very important. So getting the diagnosis, then preventing the complications in future. And now with availability of newer therapies, we can no longer say that there is no point of diagnosing genetic diseases. So I think that one myth we have to remove from our clinicians and then from the larger community, that we can't do anything for the genetic diseases. We can do a lot for them, and that is happening.
What is the one unexpected or funny moment in your lab? If you had, I'm sure you had a few.
Yeah, not funny moment, maybe not in the lab, but we have a lot of funny moments when we talk to patients actually, because many times some things which are very simple may mean something different to the patient. So we had one lady who brought her with her husband. They came and they brought their son who had intellectual disability. And then we did the test, we found the mutation. So then we were explaining कि आपके बेटे में एक जीन है जो डिफेक्टिव है. And that lady said to her husband, मैं बता रही तो इसके अंदर एक जिन बैठा हुआ है जो ये बीमारी कर रहा है. So it was really, because jinn for them means genie, like the Aladdin genie. And she was so like, it was funny, but we could not laugh there, and so then we had to calm her down. And so sometimes things, words mean different for different people, and you have these funny moments happening.
I think we need a gym. It's a really good one. So, if your life had a theme song, what would that be?
Songs.
You're making
I'm not very
yeah,
good with music as such, but I think we used to hear a lot of Raj Kapoor's songs. So one from that movie Anadi, I like that.
Which one was that?
Says, किसी की मुस्कुराहटों पे हो निसार, किसी का दर्द मिल सके तो ले उधार, किसी के दिल किसी के वास्ते हो, दिल में प्यार जीना इसी का नाम है. So
I think you have been doing that. You have been taking a lot of pain from the society and but made it.
Yeah.
So that's perfect. I think that you got, and you sing very well.
No way.
Yeah, you do. I think I'm not joking. I really, when you said I don't know songs, I don't sing, I think that was pretty good. So I think now you gave a theme song, but what would your Netflix movie be called? If you had to have one made after you and your research.
I think you have to say
Yeah.
thank you, genes.
Thank you.
Yeah, so because yeah, no, but you really have to be thankful to your genes, because we should feel ourselves so lucky that we got the normal genes. Our genome doesn't accept one difference in 3 billion. That one difference can play such a havoc that we really have to be thankful to our genes for being normal.
I think that's great. I think I really had fun, and also it was extremely informative both for the audience, clinicians, and patients. So thank you so much. I think
You enjoyed a lot
really,
talking about
really
that.
appreciate you coming here and talking about genetics, genomics, and many other things. So thank you again.